Come on. Okay. You can see my slides, correct?
Yeah.
Okay. So our last lecture is on the muscle system or the muscular system. So we've talked just a little bit about muscle tissue a few times before, particularly in our tissues lecture, and we learned that there were three types of muscle. There was skeletal muscle, cardiac muscle, and smooth muscle. When you think of muscles in your body, you are most likely thinking of skeletal muscle. This is the muscle that is connected to our bones. It's the muscle that moves our bones when we move. Smooth muscle generally is in locations where we have something that is in a circular shape. Most of those cases are going to be blood vessels and our digestive system. So this is the muscle that's going to control the diameter of our blood vessels. This is the muscle that's going to help move food through our digestive tract. And the third type is cardiac muscle. This is found only in the heart. This is the type of muscle that causes our heart to contract and pump blood. So we are going to use skeletal muscle as our primary model of muscle. Most of what we talk about is applicable to both smooth muscle and skeletal muscle, though. It's easier to picture skeletal muscle, and that's what we always think of as muscle, and so we're going to talk about this in terms of skeletal muscle. When we talk about muscle tissue, we're going to talk about it on a number of different levels. A muscle is not the same thing throughout. It is actually, what we're going to see, it's a bundle of bundles of bundles. I realize that sounds odd, but you'll actually see that there's a lot of different levels you can look at this. There's a whole muscle. The muscles are made up of little twisted muscle fibers in a fascicle, and then inside the fibers are different things, and those things are made up of smaller things, and it's very similar to our level of organization in an organism, right? Where we had the organism on the top, or I guess the organism was at the bottom of the pyramid, and atoms were at the top of the pyramid. So here, if we start at muscle, and when we talk about a muscle here, we're talking about something that you would think of as a muscle, like your bicep. It is a large, tangible thing. If you take that muscle and you cut it and you look at it, we're going to see that inside of that are going to be bundles of cells. Those cells are technically called muscle fibers. I have no idea why someone decided that they were going to call cells fibers. In my opinion, it's a stupid idea because everything in these muscles are going to look like fibers. And so you're going to take something that really isn't even a fiber and name it a fiber when the things that look like fibers aren't named fibers. It doesn't make any sense to me. But it's very important to remember, when we talk about muscle fibers, we are talking about a cell. This is not some protein. This is not some structural thing. This is an actual muscle cell when we talk about muscle fibers. So within that muscle, we have a bunch of these muscle fibers that are going to be bundled together. Muscle fibers are generally pretty long and skinny. We're going to bundle those together, and that bundle is going to be a fascicle. And we'll see these on pictures coming up. If we take one of those muscle fibers, one of those cells, and we look inside of that cell, there are going to be bundles of other fibers, what look like fibers, inside of that cell. The bundle inside of a cell is called a myofibril. That myofibril is going to be divided into sarcomeres, and the sarcomeres are going to be made up of myofilaments. And there's two types of those, there's thick and thin filaments. So here we see some of that. So on the top of our figure, you are looking at a muscle. It doesn't say what muscle this is, but let's just pretend this is your bicep. You know where your bicep is. You have a general idea of what a bicep looks like. So here we have cut it in a cross section, and you can see that within that, there are very distinct bundles of things in that muscle. Each of those bundles is called a fascicle. Now, if we pull out one of those fascicles and we zoom in on it, we are going to see individual muscle fibers. Now, I don't like part of this diagram because notice when we look at the muscle, they draw a little square and then they seemingly show us that what we're seeing in that square is the middle part of the diagram. But that is not the case. What we're seeing in the middle part of the diagram is this whole fascicle that got pulled out. It is not just what's inside this little box. It's this whole fascicle. And within that fascicle, again, we can see that there are clearly little bundles inside of that fascicle. And one of them is pulled out, showing that that thing that's in that fascicle is a muscle fiber, which remember is a muscle cell. And so the thing that is jutting out from the middle part of this diagram is a single muscle cell. And again, they draw a little box on that and seemingly tell us that we're going to zoom in on what's in that box. That is not true. The bottom part of the diagram is this whole muscle fiber. So we zoom in on that entire muscle fiber, and now we can see again that inside of that there are more bundles. Each of these bundles are going to be made up of the sarcomeres and the myofilaments. And we'll zoom in on each of those bundles on a later slide. So I want to take a second and look a little bit more at this muscle fiber. So remember, this is a cell. What you're looking at here are nuclei. Remember that skeletal muscle had multiple nuclei. Why do you think in this particular cell, those nuclei are on the outside of the cell? I mean, they're within the cell membrane, but they're still inside the cell. But they're pushed to the very edge of the cell. In most other cells, the nucleus is going to be kind of in the middle. Why do you think in this muscle cell, the nuclei might be pushed to the outside? Any ideas? So, like I said, in most cells, it would be in the middle. Why can't the nuclei be in the middle here? So that's a very good point. So he said, so they don't get squished by the contraction. Yes, that is true. What is causing that contraction? It is the sarcomeres and the myofilaments in here. And so the main purpose of that muscle is to contract. And so we want as much of that the contractile proteins in there as possible. And so the nuclei kind of get pushed away. If you were taking this in person, you were able to access the models that we have showing the muscle fibers, you would see that the nuclei actually kind of bulge out. And so that the cell membrane actually is going to bulge out around that nuclei, basically making room for it. Another thing I want to point out is this sarcolemma is the cell membrane of the muscle cell. They changed the name on us. they decided that we're going to make an entirely new name for the cell membrane of a muscle cell. And that is the sarcolemma. But it serves really no other purpose than any other cell membrane. So it's just one more thing that we have to memorize. Cell membrane of a muscle cell is called a sarcolemma. Now when we look at these muscle cells, these muscle fibers, we're going to see that it's a fairly complex structure. It has all of the normal cell parts, but there's also a lot of other things in there as well. There is the cytoplasm, but because, again, they want to make it as complicated as possible, they decided we're not going to call this cytoplasm anymore. We are going to call this sarcoplasm. So the cytoplasm in a muscle cell is called sarcoplasm. We talked about how the cell membrane or the plasma membrane is called the sarcolemma. There are going to be nuclei here. We saw them. We know that there's multiple nuclei. There's going to be mitochondria. We have to have energy. Muscle cells actually have an extremely high concentration of mitochondria. Why do you think muscle cells might have more mitochondria than other types of cells?
They need more ATP.
Yeah. Muscles are going to do a lot. They're going to provide a lot of force, and anytime you do something, you need energy. That energy comes from ATP. And so if we need a lot of energy, we need a lot of ATP. ATP is made by mitochondria, and so we're going to need a lot of mitochondria. There's also a protein in muscle cells called myoglobin. This is very closely related to hemoglobin. Remember, hemoglobin is the protein in our red blood cells that carries oxygen. Myoglobin is designed to store oxygen within muscle. Why do you think muscles have myoglobin to store oxygen, but other cells in our body do not? What is the role of oxygen? So the oxygen is used by the mitochondria to make ATP. And so you can have all the mitochondria that you want, but if you don't have enough oxygen to make the mitochondria function, they're not going to do any good. And when you need to run, if there's a rabid bear chasing you, you need to run right now. You can't wait for the blood to bring oxygen to your muscles. Your muscles have to keep their own supply of oxygen. Now, we're going to see at the end of the lecture that myoglobin only holds enough oxygen for a few seconds. After that, we have to find other ways around that. But at any given time, if we are rested, muscles are not reliant on any other part of the body in order to have the energy and the oxygen to run. They can just go. We also had those myofibrils. Those were the little bundles within a muscle cell. They're made of myofilaments, and again, we'll see those later on. The myofilaments come together. If we look at a myofibril, we can divide that myofibril into sections called sarcomeres. That'll make more sense when you see a picture of it coming up. Those myofilaments come in two flavors. We have thick filaments and we have thin filaments. Thick filaments are made of one type of protein and one protein only. That is myosin. Myosin is the protein that actually causes the contraction. It is the motor part of our muscle. So myosin protein makes up thick filaments. The thin filaments are made up of three different proteins: actin, tropomyosin, and troponin. The thick and the thin filaments are going to work together to cause the muscle to contract. The myosin is the muscle, but the thin filaments are kind of like the wheels on the car. The engine doesn't make the car go unless there are wheels on the car. And we'll see all about that coming up. We also have the sarcoplasmic reticulum, which is the same thing as the endoplasmic reticulum. In this case, it does have some extra functions though. It is going to contain something called the terminal cisterna. The terminal cisterna are going to contain a large storage amount of calcium. And we'll see that calcium is going to be very, very important in causing the muscle to contract. Finally, we have another structure called T-tubules. The T-tubules are actually little tunnels of the sarcolemma that kind of come into the middle part of the cell. And I'll point that out on a diagram when we get there. But the sarcolemma, remember, is our cell membrane. So essentially, we're going to have little parts of the cell membrane. Imagine if you took a water balloon and you poked your finger into it. You're going to take part of that membrane and you're going to make a little tunnel into the center of that water balloon. This is where sodium is going to come in. And the sodium is going to play a role with the calcium in order to cause the muscle to contract. So here again is our muscle fiber. Remember, this is a single cell that we are looking at. Within that, we have a number of myofibrils. Here we've taken one myofibril and we have zoomed in on that myofibril. Now, when we look at this myofibril, we see lots of little microscopic structures. These lines within that myofibril are the thin and thick filaments. These are the myofilaments. And if you look at a myofibril, it's very long. And within the myofibril are repeating units. One single unit is called a sarcomere. And that's what we're seeing on this bottom part of the diagram. A sarcomere runs from one Z disc to another Z disc. And I'm going to just skip ahead a couple slides to make sure. Yeah, okay. Come on. So we'll talk more about the structure of the sarcomere in a few slides. Here is that T-tubule. And so I said, imagine poking your finger into a water balloon. That's what we're seeing here. You're seeing the sarcolemma, which is the plasma membrane, and then there are these little tunnels of it that come into the cell, and they kind of wrap around a myofibril. So these are the sarcomeres buried within this T-tubule. The T-tubule is the lighter one here. Now notice that there's these two sets of three tunnels next to each other. That set of three tunnels is called a triad. A triad is made up of one T-tubule and two terminal cisterna. That terminal cisterna then has kind of this meshwork that comes off of it. That is the sarcoplasmic reticulum. So this is going to play a very important role in allowing the muscle to contract. When we start talking about how that process actually works, we're going to see that sodium is going to come into this T-tubule, and it's going to cause the calcium that is stored within the terminal cisterna to leave and go out into the sarcoplasmic reticulum. Once it's in the sarcoplasmic reticulum, then it's going to cause the muscle to contract. So looking more in depth at that sarcomere, we saw that there were lots of zones in that sarcomere. So we said that the boundary of one sarcomere, and each of these myofibrils is made up of sarcomere, next to sarcomere, next to sarcomere, next to sarcomere. And you can see that here. They've shown one complete sarcomere, but you can see where there's another one to the left and another one to the right. The boundaries between each sarcomere is a Z-disc or a Z-line. It is the same thing. It is this zigzag shape here. In between the Z-discs, we have the thin filaments and the thick filaments. The thick filaments in this diagram are the dark purple lines. The thin filaments are the green, smaller lines. If you look at these under the microscope, it literally looks like this. You can see this kind of shape. You can see that the thick filaments are bigger than the thin filaments. And you see a repeating pattern of light, dark, light, dark, light, dark. Remember that skeletal muscle, we said, was striated. And we saw pictures of that light, dark, light, dark, light, dark. You were actually seeing sarcomeres when you looked at that. Where we have this thick filament, the purple part, that is going to be dark. Between the thick filaments is going to be the light part. And so you can see how you would get light, dark out of this. Where we have thick filament, we call that the A band. Where we do not have thick filament, it's going to look lighter. We call that the I band. And so here we have the A band. If you have thick filament, you are in the A band. If you do not have thick filament, you are in the I band. Part of the A band also has thin filament. We can see that there's this little part where we have thick filament and thin filament. And part of it only has thick filament. If you only have thick filament and no light filament, you are in the H zone. If you have both, we don't have a special name for that. That is just part of the A band. And so the H zone is part of the A band, but not all of the A band. What am I missing? In the very middle of a sarcomere is the M line. Think M for middle. It is the midline. Within that sarcomere, everything is going to contract towards that midline. That midline is going to stay in place, and everything around it is going to come in towards it, bringing the Z-disc or the Z lines closer together. And if you have repeating sarcomeres, and they all get shorter, the entire muscle is going to get shorter. And so that sarcomere, remember, is made up of the thick filaments and the thin filaments. We'll see, we see on this slide what those structures look like. So on the left here, we are looking at a thick filament. A thick filament looks like a whole bunch of golf clubs that are twisted together. And so on the bottom part, you can kind of see that. Imagine taking two golf clubs, lining the heads up next to each other, and then taking the shafts and twisting them around each other. That is exactly what you're going to get here. And now imagine taking a bunch of these pairs and twisting all of those pairs together. And now you're going to get a thick filament. And so all of the shafts are lined up in the middle of that thick filament, and all of the heads are sticking out away. On the right is the thin filament. And that thin filament, remember, was made up of 3 different proteins. We had actin, we had troponin, and we had tropomyosin. So the actin is going to be the structural part of the thin filament. It is the part that's actually going to make the backbone of the thin filament. And then actin is a protein. And then if we zoom in on this, we see that that filament is made up of repeating actin. Each of these little green balls is one actin protein. And if you link them a whole bunch together, now we get a string. It's almost like a pearl necklace. And on each of those actin spheres or balls, we have one of these dark green dots. That dark green dot is where the myosin likes to grab. So remember, the myosin is the motor. That myosin is going to grab that actin and it is going to pull it. And it is going to grab it on the green dot. But notice in this picture, the green dot is covered up by this yellow spaghetti strand. That spaghetti strand is tropomyosin. Its purpose is to block myosin from grabbing the actin. And then periodically along the tropomyosin, we have these white dots. The white dots are troponin. The purpose of troponin is to control whether the tropomyosin is blocking the actin or not. And so the troponin is going to be like a light switch, an on-off light switch, either covering up the actin or exposing the actin. If the actin gets exposed, the myosin is going to grab it and pull. If troponin keeps the tropomyosin covering up the actin, the myosin can't grab it and the muscle just sits there. So, looking back at our sarcomere, are the thick filaments found in the Z disc, the I band, or the A band? Yes. So the answer is C, the A band. So the A band is where there is a thick filament. That is the definition of the A band, where there is a thick filament. Part of that A band is also known as the H zone, if you have only the thick filament. But the A band is wherever we have a thick filament. So I believe on your slide there was another question here, and I moved it. There was a couple questions that I moved to a little bit more logical place, but I didn't delete any questions, so we'll get to them. So the next thing we have to talk about is what actually causes this to happen. Now, I'm not going to go through this list right now. This is essentially a summary of the slides that are coming up. If you can walk through this sequence of events when we're finished, that is the sign that you understand how this works. Because if you can walk through this and not just regurgitate this list of events, but actually understand what is happening at each of these steps and how that affects the next step, that means that you have this down to the level that you should have this. So, muscles are controlled ultimately by our nervous system. In order for our muscle to contract, they have to receive a signal from the brain or the spinal cord telling it to contract. And so that signal is going to come through neurons. And so there's going to be a neuron coming from my spinal cord or from my brain to every muscle in my body. And so what we're seeing here is called the neuromuscular junction. This is where the nervous system and the muscular system come together, which is why it is the junction. And so we have an axon coming from a neuron, and it comes in, and then it kind of branches, and then there are little fingers that reach out and come down to the muscle. At that muscle, we have a synapse. Remember, just like at a synapse between two neurons, between a axon and a dendrite, they don't actually touch. We're going to have to use neurotransmitters to send a signal across that synapse. And so, an action potential comes down this axon. When it reaches the end of the axon, it causes the neurotransmitter, which in this case is acetylcholine, to be released into the synapse. That neurotransmitter, that acetylcholine, then floats across the synapse, and it is going to bind to receptors on the muscle. Remember when we talked about action potentials in neurons, the neurotransmitter caused a sodium channel to open, which caused sodium to rush in, which created an action potential in the next neuron. The same thing is going to happen here. It's just instead of creating an action potential, it's going to cause the muscle to contract. And so, the acetylcholine floats across the synapse, binds to a sodium channel, and causes it to open, allowing sodium to enter having a brain fart. What was the name of that structure? Boy, sorry about that. We talked about it. T-tubule. Okay, yes. So, the channel opens up, sodium rushes into the T-tubule. Now, if I go back to that slide I was just looking at, imagine we now have sodium that is in this T-tubule. The calcium lives in the terminal cisterna right next to it. Now, can you tell me what charge does sodium have? Positive. Can you tell me what charge does calcium have?
Positive too.
It is positive, yeah. So I don't know which you meant, but you could interpret what you just said positive two ways, and they both be correct. It is positive also, but it also has a 2 positive charge. The fact that it has a 2 positive charge isn't all that important to us, but what matters is that it is positive also. What can you tell me happens when you have a positive next to a positive? They repel, yes. Positive and negative attract. 2 positives will repel. So what do you think is going to happen if the calcium is in the terminal cisterna and we bring a whole bunch of positive sodium into the T-tubule? They're going to repel, which is going to cause the calcium to leave the terminal cisterna and come out into this meshwork of the sarcoplasmic reticulum. That sarcoplasmic reticulum, though, is leaky. So the calcium goes out into that sarcoplasmic reticulum, and the calcium is then going to leak out of it and get into the myofibril, the sarcomere, the thick and thin filaments that it's wrapped around. And so now we have calcium that can interact with that sarcomere and the thin and thick filaments. Now remember, I said that troponin controlled the location of tropomyosin and determined whether it blocked this whole contraction from happening or not But I didn't say what controlled troponin. What controls troponin is actually the calcium. When troponin does not have calcium, it leaves tropomyosin in place and it blocks the contraction from happening. But when that calcium leaves the terminal cisterna, goes into the sarcoplasmic reticulum, and then leaks out, that calcium is going to bind to the troponin. When the calcium binds to the troponin, that's like flipping the light switch. That causes the troponin to move the tropomyosin, exposing the actin. And as soon as you expose that actin, the myosin is going to jump at its chance. The head of those golf clubs is going to reach up, it's going to grab it, and it is going to pull. And as long as you leave that actin exposed, it is just going to keep doing that over and over and over again, continuing to contract that muscle. So, what was it that caused the terminal cisterna to release that calcium? So,
Is it the troponin?
So, how did this whole thing start?
ATP.
It was the action potential coming down the neuron. So the action potential came down the neuron, caused release of the acetylcholine, which allowed the sodium in, which caused the calcium to move. And so the action potential didn't directly cause the release of calcium, but none of these other things had any role whatsoever. So the ATP is going to be used by the myosin to pull, which is after the calcium left the terminal cisterna. The troponin reacts to the calcium, which again is after the release. We didn't talk about calcium pumps. The calcium pumps actually exist to shut off the muscle contraction. The calcium pumps are going to take the calcium that is in the muscle and pump it back into the terminal cisterna. And so we're going to remove the calcium, which is going to cause the troponin to put the tropomyosin back in place and block that contraction. And so when we are contracting, this is a sequential process. And so we have calcium. It has bound troponin. The troponin has caused tropomyosin to move. That when the tropomyosin moved, it exposed the actin. The myosin head is going to reach up. It is going to grab that myosin. When it does that, it is then going to pull. After it pulls, a molecule of ATP is going to bind. When the ATP binds, it's actually going to cause the myosin to let go. After it lets go, it's going to use the energy from that ATP to reach up and grab again. When you use the energy from ATP, remember ATP stands for adenosine triphosphate. There are 3 phosphates on it. If you use one of them to get energy out, now you have ADP, adenosine diphosphate. And so triphosphate causes it to release. Diphosphate causes it to grab and pull. And so we get this sequential process of it reaches up, it pulls, ATP binds, we let go, we break down ATP to make ADP, and then we reach up, we grab it again, and then we repeat over and over and over again until we either reverse the process by getting rid of the calcium, or if we run out of ATP. If we run out of ATP, it doesn't matter what's going to happen. Now, I want you to think about this process. Imagine a situation where you ran out of ATP. in what state would the muscle get stuck? Would it get stuck in the relaxed state, or would it get relaxed in the activated, contracted state? So contracted, yes. You would get stuck in the contracted state. Can you think of a situation where muscle gets stuck in a contracted state?
Muscle cramps.
So you both gave me very good answers that answered my question, but unfortunately my question was poorly worded, and so they're not what I was looking for. How do I get you to what I was looking for? Well, can you come up with a situation other than... There you go. Rigor mortis. When animals die, they turn stiff, right? That's because of this. If we die, we no longer stop our breathing. If we're no longer breathing, our mitochondria stop functioning. Mitochondria stop functioning means we stop making ATP. If we stop making ATP, the muscles are going to run out of ATP. Now, this might be kind of a brain-shattering event to you, but when you die, your muscles are not dead. What we think of as death is really a made-up definition. I mean, what is death? I mean, there is the death of a cell, and then there is sort of this spiritual death, right? But we tend to think of death as being loss of consciousness. But what is consciousness, right? If back in the day when the French were big on decapitating people with the guillotine, there was a scientist that got sentenced to death, and he said, I want to know what's going to happen. How long do we survive after our head is cut off? And so he said that after my head is cut off, I am going to continue to blink my eyes for as long as I am conscious. And so he did this, and after his head got cut off, he continued to blink for like 30 seconds. The problem is we don't know, was he consciously blinking his eyes, or were they just kind of twitching? We don't know. But ultimately, the cells in his brain were not dead when the head got cut off. The cells that made up the muscle of his eyelids weren't dead. They didn't know that his head had been cut off. They still have oxygen. They still have ATP. They're going to continue on like nothing ever happened. They are going to die when they run out of ATP. And so they're going to continue to function. They're going to contract. But eventually they're going to run out of ATP, and then they're going to stay contracted. But eventually, once they're dead long enough, now the proteins are going to start to degrade, and the muscle is going to kind of relax, simply because the myosin is no longer structurally sound enough to keep it contracted. Okay, sorry. I went off on a death tangent there. So, in a resting muscle cell, the sites on actin where the myosin binds are blocked by which protein? Troponin, titin, or tropomyosin? C, tropomyosin is correct. That reminds me, we had titin on one of our other slides, and I forgot to mention it. So the titin protein is called titin because it is a very large protein, and its function is to connect that sarcomere to the cell membrane. So imagine we have this muscle cell, and then within that cell we have the sarcomeres. If the sarcomere isn't connected to the plasma membrane, The cell itself is just going to sit there, and then the things inside of it are going to contract and relax. We need to link the outside of the cell to the inside of the cell. And that's what Titan does. So I forgot to bring my phone with me. Can somebody tell me what time it is?
It says 6:58.
Perfect time for a break. So we will come back in 10 minutes. I think we got a little ways to go, and so we'll take a 10-minute break. What side am I on? 17. Oh yeah. Let's take a 10-minute break. We will come back at 7:08. Okay, so we're going to pick up where we just left. And so we went through the whole process of the action potential coming down, ultimately causing the sarcomere to contract. When we talk about that sarcomere contracting, we talked about how the thick filament was going to reach up and grab the thin filament and pull it. But if you zoom out one level and look at how this works in a single sarcomere, we get what we call the sliding filament mechanism. And so we see a sarcomere here. We have the M line running down the middle. We have the thick filaments. The thick filaments are actually going to stay in place. They're going to stay in one place. They're going to reach up, grab the thin filaments, and pull them towards the M line. And so imagine my fingers are the thin filaments, and then in between them here, we have thick filaments. Essentially what's going to happen is the thick filaments are going to pull them together until they reach the M line. And so the sarcomere comes together and causes, if it gets shorter, like I said before, if all of the sarcomeres get shorter, the entire myofibril is going to get shorter, and that myofibril is tied to the cell membrane of the cell. Sorry, I got sneeze. That was kind of a wimpy sneeze. Okay, so then the cell gets shorter. The muscle itself is made up of a whole bunch of cells, and so if all of the cells get shorter, then the muscle itself is going to get shorter, which is going to cause the muscle to contract. We talked about the relaxation. We said that this relaxation is caused by the calcium getting pumped back into the sarcoplasmic reticulum. So now, if we look at how these protein contractions influence the contraction of a muscle, we can make a muscle twitch graph. So what you're looking at here, on the x-axis, we have time measured in milliseconds. A millisecond is 1/10,000th of a second. Sorry, why did I say 10,000? A millisecond is 1,000th of a second. And so 100 milliseconds, which is the end of our graph, is a tenth of a second. And so a millisecond is sort of a non-relatable amount of time, but a tenth of a second is something that you can relate to. And so what we're looking at is the amount of force being applied by this muscle over a time span of a tenth of a second. And so we see that it is at the beginning, it is released. It has no force, it is relaxed. Then we get this whole process that kicks in, and it starts to apply a whole bunch of force, and then the muscle is going to relax. Notice that there's a delay here. The delay, that zero time point, is when the action potential got to the neuromuscular junction. Why do you think there is a delay between the action potential reaching the muscle and the muscle contracting?
Because it needs time to reach the muscle.
So yes and no. And so the action potential, the zero time point is when the action potential reaches the muscle.
Oh.
But there were a number of steps that had to happen, right? The acetylcholine has to be released. It has to get across the synapse. It has to bind to the sodium channel. The sodium has to come in. The calcium has to leave. The calcium has to leak in. It has to bind to the troponin. The troponin has to move the tropomyosin, and then the myosin can grab the actin. All of that happens in this tiny little latent period. And so it is very short, but there is actually a delay between when that action potential reaches the muscle and when the muscle contracts. Now, it contracts very, very quickly because that calcium just floods out of that sarcoplasmic reticulum into the muscle, binds the troponin, and the contraction happens. The relaxation period takes much longer than the contraction period. Why do you think that is? Why would it take longer to relax than to contract? Think about what is causing the relaxation.
Because it's harder to get rid of the calcium ions than it is to force them in.
Yes and no. So, like he said, sodium pumps have to pump sodium. Calcium pumps also have to pump calcium. So we have to pump the calcium from the sarcoplasm into the sarcoplasmic reticulum and back into the terminal cisterna. We also have to pump the sodium back out, because if the sodium is still there, when we pump the calcium back in, the calcium's just going to want to leave again. So we have to pump the sodium out, we have to pump the calcium back into the terminal cisterna, and that takes time. That is this relaxation period. However, we do not have to wait for that relaxation period to completely finish before we contract again. If we don't wait for the relaxation to finish before we contract, we undergo what's called wave summation or temporal summation. Wave summation because it looks like a wave. Temporal, the word temporal always refers to time. And so here is, we are going to, over time, add the force of multiple contractions on top of each other. And so we see each of these peaks is a separate contraction. And so you can imagine, if we contracted once and then we waited long enough, eventually it's going to get back down to zero, and we're just going to go up and down, and up and down, and up and down, and every peak is going to be the same height. But if we don't completely relax before we contract, every time we contract, we're going to go the same amount higher than where we started at the beginning of that contraction. But if we didn't make it all the way down to zero, the second contraction is going to go higher than the first one. And the third one's going to go higher than the second one. And the fourth one's going to go higher than the third one. That is wave or temporal summation. Summation means summing, or the sum is addition, right? So in this situation, we are adding multiple contractions on top of each other to increase the strength of that contraction. And so if I pick something up and I need to apply more force to it, I am going to contract faster. As the muscles contract faster, we get more of this summation going on. And so we are able to apply more tension and more force. If these contractions happen very, very, very close together in a very short period of time, if we kind of zoom out on the graph, rather than seeing this wave form, we essentially see a straight line that comes all the way up and then plateaus. This is called tetanus. Tetanus is the state of muscles being at maximum contraction and staying there. What do you know the word tetanus is meaning? Where do you know the word tetanus from? Yeah, you get a tetanus shot. Do you know what that tetanus shot does? What is it?
vaccine.
It's a vaccine. Do you know what it's a vaccine to? Vaccines prevent infections, right? Well, what are we preventing?
Is it the tetanus toxin?
So tetanus toxin comes from a bacteria. So we're preventing the infection, the bacterial infection, because if we prevent the bacterial infection, then we can't get enough bacteria in order to create enough that toxin. That toxin causes this muscle contraction, very strong muscle contraction, which puts our body in a state of tetanus. Tetanus is a symptom of this infection. Now, to take another step back, where do we get this infection from?
Like stepping on rusty nails and...
Yeah. And so a lot of people would think we get tetanus from rusty nails. It has something to do with the rusty nail. That's actually not the case. The rusty nail does not hurt us. Well, if we step on a rusty nail, that is going to injure us. But it's actually the bacteria that causes tetanus likes to live on rusty metal. It will also, somebody said you got a tetanus shot for a cat bite because it can also live in a cat's mouth. And so the cat bites you, they puncture the skin, they essentially inject that tetanus the bacteria into your bloodstream, where you then get an infection. The infection grows, releases a bunch of toxin, and you go into a state of tetanus. Similarly, so this graph looks very similar to our wave summation graph. However, we have weighted between each contraction until we get down to 0. We call this tree. Essentially, each peak is almost the same height as the one before it. Muscles get a little bit more efficient when you can cause them to contract over and over and over again. And so the second contraction is going to be just a little bit stronger than the first one. You're going to go all the way down to 0 between, but the second one's going to come up just a little bit higher. And the third one's going to be a little bit higher. And the fourth one's going to be a little bit higher. But notice that we're not getting nearly as strong here as we did with our wave summation. The wave summation is going to be needed in order to get a very strong muscle pull. And so, when this happens, remember what this is actually causing. Sorry, I got an eyelash in my eye. This is causing the muscle to get shorter. And I like this diagram because it does a very good job of showing how a muscle getting shorter causes our body to move. And so you're looking at an elbow here, and you're looking at the bicep that is on the front of your upper arm. And you can see that one end of that bicep is attached to your shoulder. The other is attached to your forearm, just below your elbow. Now I want you to look, extend your arm, and look. And imagine if I have a muscle connecting my shoulder to a point just below my elbow, and that muscle contracts, it's going to pull those 2 points closer together, right? If the muscle connecting those 2 points gets shorter, I have no choice but for those 2 points to get closer to each other. And the only way for that to happen is to go like that. And so when my bicep contracts, the point on my forearm has to get closer to my shoulder. When we do that, that is called a concentric contraction. That's a contraction that is bringing one point closer to the other. if we are going to, we are going to do that. In the picture, we show a picture of somebody lifting a weight. If we are going to let that weight down in a controlled manner, so I'm going to set that down, I can't simply completely relax my arm all at once. That's just going to make my arm just flop down. And that's not what we're trying to do. We have to maintain some contraction while letting the weight down. That is called an eccentric contraction. A concentric contraction brings the 2 points together. An eccentric contraction is a contraction that occurs while we are taking them further apart. An isometric contraction is a contraction that occurs in our muscle in order to keep it in a set position. So we see a picture of somebody that picked the weight up a little bit and they're holding it there. Now, if you've ever tried to lift something up and just hold it there, you know that actually takes a surprising amount of force. If you've never done it, just take a gallon of water or something and pick it up. It's not heavy, right? Just pick it up a little bit and hold it. And hold it, and hold it, and hold it, and eventually you'll realize that gallon of water is surprisingly heavy in your muscle. Your arm is going to start to shake, and you're going to get very uncomfortable, and you're going to have to put it down. This is the same idea of if you lay on your back and you lift your ankles you lift your feet just a little bit off the ground. It's really easy, really simple, but if you try to hold it there, you're going to realize you don't have the muscle tone that you thought you did. That is an isometric contraction. The prefix iso always refers to same. We saw this with isotonic. We had two solutions that were the same concentration. Metric, this is we're talking about think of a measurement, right? Same measurement, same length. This is a contraction that's needed to hold an object in place. So we have concentric contraction, which moves it up, an eccentric contraction, which moves it down, and an isometric contraction, which maintains it in one place. So now we're going to stay in still the muscle lecture, but this last material comes from a different chapter of your book. I'm going to talk more about the broad functions of the muscular system and how these muscles work together to have an effect. And so we have four primary functions of the muscular system. There's movement. It allows me to move from one place to another. It allows me to move my arm up and over and do whatever I need to do. The muscles are going to allow me to maintain my posture. It allows me to sit upright. Gravity is trying to pull me down. I'm continuously using isometric contractions. These are what we tend to refer to as our core muscles, right? They keep us upright. If they were to simply relax and let go, I would fall over. Muscles are going to stabilize joints. When we saw the picture of our knee last week, we saw that there are muscles around it. They're going to help hold the bones upright, stabilize those joints, prevent them from slipping and falling over. And finally, we are warm-blooded animals. We need to maintain 98.6 degree core temperature. It is not always 98.6 degrees outside. If it is colder, it is not 98.6 degrees in this room that I am in right now. In fact, it is in the 70s, yet I had to turn the ceiling fan on because my muscles continuously generate heat. As I'm talking to you, the muscles that are allowing me to talk to you are generating heat. As I wave my hands, I'm generating heat. As my diaphragm moves, I am generating heat. Think of it like the engine in your car. whether that engine is a gasoline engine or a diesel engine or an electric engine, it creates heat. It is a byproduct of the energy of the movement process. One thing we didn't talk about, so this is the same diagram we looked at before, and I completely ignored some of the terms on here: the epimecium, the perimecium, and the endomecium. These are layers of connective tissue that kind of wrap around some of these layers of organization and act as protective layers and to help us kind of define the boundaries between bundles. So if we have a muscle, so remember we said this was as an example of like a bicep. This is an entire muscle. Wrapped around that muscle is a connective tissue layer called the epimecium. Epi always refers to outside. So this is the epimecium, is on the outside. Within that muscle, we said we had muscle fascicles. The fascicle itself is wrapped with perimecium. The perimecium is wrapped around one fascicle. Within that one fascicle, we have multiple muscle fibers. That muscle fiber is wrapped in endomecium. Endo refers to in. So epimecium is on the outside, endomecium is on the inside, and the perimecium is in between. You're going to want to know what each of these different connective tissue layers wraps around. So the epimecium wraps around a muscle, the perimecium wraps around a fascicle, and the endomecium wraps around a single muscle fiber. And so that muscle fiber itself has the sarcolemma or the plasma membrane around it, but then there's another layer around that, which is the endomecium.
So the endomecium is underneath the sarcolemma.
It is on the outside. The endomecium is on the outside of the cell. So you have endomecium, and then inside of that is the sarcolemma or the plasma membrane. So if we look at muscles, we have a number of different terms that we apply to muscles and pairs of muscles. So when we were looking at that bicep, we were connected in two places. We said we were connected at the shoulder and we were connected to the forearm. Skeletal muscles are going to have two ends. They're going to have two attachments. And when we contract that muscle, one end is going to stay stationary, the other is going to move. And so when I contract my bicep, my forearm moves very clearly and my shoulder does not. The end of the muscle that does not move, we call the origin. The end of the muscle that moves is called the insertion. So we have two ends. We have a stationary end. We have a moving end. The end that stays in place is the origin. The one that moves is the insertion. In most cases, skeletal muscles come in pairs. If my muscle goes if I want to move my body this way, I also need to move my body that way. And so there's going to be one muscle that moves it this way. That's my bicep. There's going to be a muscle that moves it this way. That is my tricep. When we talk about a muscle that is going to cause a joint to flex, that is the prime mover. So the bicep is the prime mover of my elbow. We also call that the agonist. The muscle that has the opposite effect of the agonist is the antagonist. And so the antagonist is my tricep. A synergist is a muscle that is there in order to help the agonist. So before we do that question, here's an example of the prime movers and the synergist. And so we said the prime mover was the bicep. But there are a couple of other muscles that help it. We have the brachioradialis and the brachialis. These are two muscles. One of them is in the upper arm. The other one is in the forearm. But they both act to help us flex our elbow. However, the dominant muscle here is the bicep. So the bicep is the agonist. The other two are synergists. And so that's what we just talked about. The answer to this one is the prime mover or the agonist. So I think this is the last topic, correct? Yeah. So the last thing we have to talk about is where we get the ATP from to make all of this magic happen. Now, we said it comes from the mitochondria, which is true. But the mitochondria need fuel. They need glucose and they need oxygen. And so the question is, where are we going to get that from when we need to run from our rabid bear? We have two ways in order to make ATP. One of them is the mitochondria. There is also a way to make ATP without mitochondria. So the mitochondria are the preferred way to make ATP. This is aerobic respiration, the second bullet point here. Aerobic refers to using oxygen. This makes far more ATP. We get 38 ATPs when all is said and done. This is a multi-step process, but when all is said and done, when we can convert 1 molecule of glucose into 38 ATP molecules. Not only that, but we get essentially no toxic byproducts from that process. The downfall of it is we need oxygen to do that. But our muscles don't always have enough oxygen to do that. And so when our muscles run out of oxygen, we're going to switch from aerobic respiration to anaerobic fermentation. This allows us to make ATP without oxygen. However, instead of 38 ATPs per glucose molecule, we get 2. It's an incredibly inefficient process. We don't get nearly as much ATP using anaerobic fermentation as we do aerobic respiration. On top of that, we make lactic acid using anaerobic fermentation. Lactic acid is going to cause cramps. When you get a cramp, this is primarily due to a buildup of lactic acid. It's also going to cause your muscles to get tired. However, in a lot of situations, we don't have a choice. We have to go and switch to anaerobic fermentation. And so where our body is getting this energy from is going to change depending on how long we have been running. So when you first start running, we have oxygen. Remember, we store oxygen on the myoglobin, which is inside of the muscle cell. The muscle cell has its own store of oxygen. So when you start running, you can use aerobic respiration all you want. You make a whole bunch of ATP. However, After about 10 seconds, you run out of oxygen. 10 seconds, that is not very long. After that, we're going to turn to what is called the phosphagen system. We have a slide about that. I think it's the next slide. This is going to allow us to make ATP without actually using aerobic respiration or anaerobic fermentation. There's going to be a quick little brief time period where we don't have to use anaerobic fermentation. Essentially, the phosphagen system is going to be a bank where we can store up ATP. Once the phosphagen system runs out, now at the kind of 20-second time period, we're going to have to switch over to anaerobic fermentation. We're going to use anaerobic fermentation for as long as we continue to run. However, once we stop running, we are going to have to repay that debt. We call that oxygen debt. That is why when you stop running, even though your muscles are no longer contracting, you continue to breathe heavily. We made all of this lactic acid. We can't let that lactic acid stay there. What we need to do is we need to get oxygen and then kind of use that oxygen to get rid of the lactic acid. So we went into debt. We say, I'm going to continue to run and burn glucose in the absence of oxygen, but at some point I am going to have to repay that debt and get that oxygen into my body. So you're going to continue to breathe hard until that lactic acid has gone. So we said, for short exercise, for about 10 seconds, we have enough oxygen on our myoglobin, but then we have to switch to that phosphagen system. The phosphagen system is going to allow us to take phosphate groups from other molecules in the cell to make ATP. Remember, ATP, adenosine triphosphate, 3 phosphates. And we get the energy out of the ATP by cutting one of them off. When you cut one of them off, you now have ADP, adenosine diphosphate. And so we need to find a phosphate somewhere that we can stick on to our ADP in order to convert it back into ATP. We have 2 ways of doing this: myokinase and creatine kinase. Myokinase says, well, I have plenty of ADP floating around, right? Why don't I just take one of the phosphates off one ADP and stick it onto one of the other ADPs? I'll then have an ATP and an AMP, adenosine monophosphate. Problem solved, right? ADP doesn't do me any good. AMP isn't any worse than ADP, so let me just convert an ADP, take part of it off, and recycle part of it. Creatine kinase is going to take a phosphate off another protein called creatine. Creatine phosphate is essentially a storage form of phosphate in our muscle cells. We're not working, and we have plenty of ATP. Our body can say, well, I'm going to store some of this ATP. I'm going to take a phosphate off of an ATP, and I'm going to stick it on creatine. And then later on, when I need that phosphate, I'll come get it off the creatine, stick it back on ADP to make ATP. These 2 things together make up the phosphagen system. So this together, the phosphagen system gives us about 6 seconds. And so the myoglobin gave us about 10. The phosphagen system gives us about 6. And so this slide is making the assumption that we can run the 100-meter dash in about 16 seconds. The first 10 seconds are going to be the oxygen with the myoglobin. The last 6 seconds are going to be the phosphagen system. Here we see the myokinase and the creatine kinase. So these are enzymes. Remember, enzymes are proteins that do something. They cause a chemical reaction to happen. And so these arrows are showing a chemical reaction. And so we have myokinase in the middle. We have two ADPs that come in. The myokinase takes a phosphate, the P, off of one of the ADPs, sticks it on the other one, and we end up with an AMP and an ATP. The creatine kinase takes a phosphate off of the creatine phosphate, sticks it on ATP, and we end up with creatine and ATP. So does that phosphagen system make sense? Do you have questions about the phosphagen system or any of the energy systems we've talked about so far? Okay. When the phosphagen system is gone, then we have to convert and move over to the anaerobic fermentation. This, we said, allows muscles to use glucose, but in this situation, they're not reliant on the circulatory system to get them extra oxygen, because the circulatory system cannot get them oxygen as fast as they need it. And so the muscles are going to use glucose. Not only do they have glucose, and not only are they getting more glucose from the bloodstream, but muscles have some glycogen in them. And remember that glycogen was a storage form of glucose. We take a whole bunch of glucose molecules, we string them together, and we get glycogen. And so muscles can have glycogen in them. And so when we start running, the muscles are going to very quickly use up the glucose they have sitting around. And then they have to go to their bank, and they have to start breaking down the glycogen to get the glucose out. But when we do that, we get lactic acid, and then we have to get rid of the lactic acid. We have enough glycogen in our body to let us run for about 30 to 40 seconds. And so between the myoglobin, the phosphagen system, the 30 to 40 seconds of the anaerobic fermentation, we have enough to run about a minute. But we can run more than a minute. So what do you think is happening? What's going on? It seems like to me that we just talked about that we only have enough oxygen to run for about a minute. What's going on? Let me highlight. I got this is a slide. I can't highlight it. It says maximum activity. What's the importance of that term, maximum activity?
It's like using all of your resources.
Yeah. So this is sprinting. We can sprint for about a minute. Well, theoretically, we can sprint for a minute. I'm not going to claim that I can sprint for a minute. But in theory, we can sprint for a minute. But the idea is even athletes cannot sprint for more than a minute. We can jog for a long period of time. Because at that point, we are able to replenish our muscles with oxygen and glucose faster than we are using them. And so an athlete is able to continue to run basically indefinitely, right? However, in that situation, their circulatory system, their respiratory system is in very good shape. And so they're able to supply more oxygen and more glucose to their muscles than the ordinary person. The ordinary person cannot jog indefinitely. Eventually, we get too far behind and we have to stop. Their muscles are also healthier. And so they are able to be more efficient. They can get more movement out of less ATP, and they're able to get rid of the lactic acid better. So somebody asked, does this involve fast twitch or slow twitch muscles? Generally, these are going to be slow twitch muscles. If we're normal movement is going to be a slow twitch muscle. So this slide is a reminder that for the lab exam and for the final exam, you are responsible for knowing the muscles, the muscle names, the muscle locations, and the muscle actions of the muscles that are in your lab manual. For lecture exam 4, I am not going to ask you what is the function of the brachioradialis muscle, or where is the brachioradialis muscle found. To me, that is not lecture material. That is lab material. You will have questions just like that in the lab exam. And the final exam covers lecture and lab. And so you will need to know it for the final exam also. So that's all this slide is. So that is the end of our muscle lecture. Do you have any questions about the muscles? Specifically about the muscle lecture? Okay. So if there's no questions about the muscle lecture, are there any last minute questions about lecture exam 4 or the lab exam or the jello? All right. So remember, they are due on Monday. If you're watching this recording, it's probably not due on a Monday for you. So somebody asked, will the final exam have diagrams? There are one or two. There are not a lot of them. It's not like the final exam where there's a whole bunch of them. But there are one or two diagrams on the final exam. They're fairly simple diagrams. They're not complicated, but they are there. Anything else? Is the final exam 100% new questions? The answer is yes. I am not going to guarantee that there is not maybe, possibly one or two questions that you've seen, but if they're there, it's a coincidence. Because I made my exams, the department made the final exam. It's possible there's an overlap. I don't think there is. I don't remember there being any, but I'm not going to tell you that there aren't any questions on both. Anything else? So we will have the final exam review next week. And so you want to start on that. I would suggest not completely waiting until after you do all these other exams to start studying for the final. And I would also suggest taking the other exams as soon as you can. Not only because you don't want to wait till the last minute and then have something go wrong, but that also gives you more time for the final. And remember, the final is a beast. It is 20% of your final grade. It is the most difficult exam of the semester. Do not overlook it and give yourself as much time as possible to take it. That said, don't rush through the other exams. Study for them. Don't procrastinate studying for them. Study for them, take them, do well on them, and then move on. So if there's nothing else, I will see you